So my background is in statistics and genetics. I've done a lot of work in methodology for AI, and I thought maybe I could talk about that, but it doesn't feel quite connected to the actual topic. I couldn't see whether it was going to be useful. So instead of talking about that, all my work in the ICU data with the local NHS, which also just didn't get beyond the let's try to get at the data and run very simple data science analysis on, I figured I would talk about my genetics work, which, whilst not truly being AI, at least should be in the bin of data that gets put in there. So this is about local ancestry. Does it matter for CHD? So and I should thank the people that have been working on this. So I guess the people that are at Bristol, Yelling's PhD student who's just finished. Amy is at the IU and works at the last section. And then the other people from around the country have made very important contributions too. Good. So the first thing I thought when being invited to speak here was, is anything I do relevant to CHD? And it's not entirely obvious from the outsider's perspective whether genetics needs any thought, or whether we can do very straightforward vanilla stuff and get the right answer for CHD. So I did a small literature review. I don't know whether I found the right things, but I've definitely found things. And so we have this situation that's saying there's something like 400 genes that are involved in congenital heart disease, and we're not sure about half of them. We can say that for many of them, they're sort of known and simple, but actually slightly over half, there is a complicated genetic relationship between the genome and what happens. So this tells me that there's still something to study, and indeed, when you look at what the situation is with large genome-wide association studies, we find that if you compare white people to non-white people, we get massive disparity in how well we can predict whether somebody gets CHD. And this is a massive red flag for us not truly understanding which loci are actually causal, and only knowing essentially the tagging structure in the genome. So this tells me that the problem is very, very far from solved, and that there's some interesting relationship with population structure, at least, and many of the other things that come up in genome-wide association studies. So then I thought, well, I'm an expert in population genetics. Is that relevant? So this is essentially describing which populations an individual might have genetic ancestry from. So it can be that you're simply from one population, but you might be from multiple. And so, well, we've got lots of good software to do that. We can find for a particular bit of the DNA, where does it come from? And we can do that at very fine scales, and we can do it for very, very large data sets. So that is potentially very useful, and so then I thought, well, is it going to be useful for congenital heart disease? And so just to give a scale of like how good we are at telling where someone's from, if somebody has 4 grandparents from let's say South Yorkshire, this is the distribution of where we think that they'd be from, right? So it's extremely accurate. You can get the county level within the UK right. And you can do the same thing worldwide. And this pipeline we've got sitting there, we can just throw everybody in the UK Biobank or from some other data set through it, and we can get this distribution of their genome. I don't want to go into the details of what this shows, but it's basically saying we can do fine scale attribution of where someone's ancestry is from. But what the work that I've done in this space of the last 5 years has tended to show is mostly that it's not that important. So it does help. So for example, if you try to predict someone's birthplace, try and learn which genes are associated with birthplace, specifically SNPs, single nucleotide polymorphism. You find that if you do things with kind of the old-fashioned methods of using broad brush correction for population structure, you get all of the SNPs on the genome that are associated with where you're born, and obviously that's not a causal signal, right? So you know you're getting confounding if you find this. Whereas if we use this method that corrects for local ancestry, you can get rid of most of it. And I don't know why it's come back. Sorry, just a second. Good. So yeah, we got rid of nearly all of them, and only 2 remain. So and those 2 turn out to be related to asthma. So they might be actually causal because people might be moving to get away from places where they suffer from asthma, perhaps. Or perhaps they're still false positives, I don't know. But then it doesn't really work for anything that's like very biological. So it works for something like employment score, but that's a very strange sort of phenotype. And it doesn't work, well, it doesn't make much of a difference for something like waist circumference. There's these 4 SNPs on the genome that we think were previously found and probably are false positives, but that's like 4 SNPs. It's not a big deal. So all of that says it's not that important. And when we attribute a particular variant on the genome to a particular ancestry and ask, can we understand how the effect size of that SNP varies depending on whether it's found on an African segment or on a European segment of DNA? We find that basically the only effect that's important here for pretty much all of the traits that you could think of is something called linkage disequilibrium. So there's some causal SNPs, and we don't know where they are, but essentially all we're doing when we find an association is tagging those causal SNPs, and the only thing that's changed in these 2 backgrounds is which causal SNP, what's the frequency of those causal SNPs on those 2 backgrounds. And that's the only thing that matters. So basically it's saying biologically, it doesn't matter where you're from in the world, biology hasn't changed, which is really good because it means that everything that we're learning about the genome will be true, but it's bad because we're still bad at predicting what will happen in people who aren't from Europe. So that's a challenge. And I guess more recent work has been doing this computationally efficiently, and we can do it for hundreds of thousands or millions of people, and it's fun, but not relevant to CHD. So I'm not going to go into the details. But it's very easy to tell where someone's from. So this new pipeline doesn't even need us to do any sort of supervision at all. So the previous pipeline needed 4 grandparents from a particular location. Here, we don't have that at all. We're just able to tell where someone is born in the UK Biobank from inside a typical radius of that white circle, right, which is about 40 kilometers. So that's interesting, I think, but maybe not directly useful. And so there's lots of interesting things in this data, such as looking for bits of the genome that have been under selection recently. So if you look in somebody who's, let's say, from Eastern Europe, you might find bits from Western Europe that are overrepresented and things like that. And we find them, there's lots of them, but they're not going to be relevant to CHD because they are essentially driven by immune responses. So what's been under recent selection in humans is pretty much the immune system and not much else. So when trying to attribute what's really going on, you need to find the causal SNPs. Causal SNPs are often very rare. So rare variants are very important, and people in the IEU have been looking at those for a long time. The most important design that doesn't have this problem of population structure, which is the thing that I've just been talking about, and that I'm claiming doesn't solve all of the problems, is this thing called sibling design. So it's saying if you want to know which SNPs are associated with a disease, you need to have somebody with that disease, and you need the same genetic data from a sibling. And if you've got that, then we can start to do some really serious attribution of which of the SNPs that are really involved. But without it, I don't think it's going to turn out to be a solvable problem. This is a plot from that paper that Amy has generated, and essentially all she's doing is looking at the correlation between what does the effect size look like in a population where you don't know about siblings versus if you do it with siblings. And what you find is that essentially it's not very good. That's the point, right? So you'll not get the right answer if you're only looking at population level statistics. You need these siblings. She was looking at rare variants, and this is the example of something where a rare variant is very geographically isolated and is a really good candidate for telling you that there should be some correlation with the population structure and this rare variant, and even in this case, it still doesn't work to try to correct for population stratification. You only get the right answer if you use the siblings. So, basically that's all there is to say. We really need access to sibling level data in order to be able to get really good attributions, of which SNPs are causal, and that's really important for all of the downstream tasks, things like figuring out what's going on with the metabolomics and all of that, and the causes of the disease. Whether that's useful for treating the disease is also completely unclear, I think, at this point. There's not that many cases where knowing about the disease, the genetics behind the disease has been that useful. But I still think it's interesting, and we should definitely still be doing it. So that's the conclusion.